Imagine waking up one morning and not being able to remember the face of the person you love most. As your mind slowly fades away, what could be more terrifying than losing everything you have, your memories? Now, imagine that somewhere on the forest floor right now, probably within a few miles of wherever you're sitting, there is a mushroom. A weird, almost disturbing-looking mushroom that oozes what appears to be blood. And that mushroom might hold a key to one of the most devastating diseases in human history. I'm not exaggerating. I want to be clear about that right off the bat. I'm not here to sell you supplements or tell you we've cured Alzheimer's. But the story of this mushroom, Hydnellum peckii, the bleeding tooth fungus, is one of the most genuinely fascinating things I've come across in years of poking around on forest floors. And I've come across a lot of weird things on forest floors. Hello, I am a 29-year-old researcher with a great interest in mushrooms, and I also make amateur documentaries on the subject. I've been obsessed with mushrooms since I was 22. Back then, a documentary I watched about mushrooms significantly changed my perspective on life, nature, and people. I won't go into details. That documentary pushed me into a 3-year obsessive self-learning journey in mycology, and here we are. I live outside Eugene, Oregon. If you didn't know, this place is considered the mushroom capital of North America. I'm serious. When it rains here in the fall, and as you know, it rains a lot, the forest floor transforms into something that seems like it belongs to another world. Almost every weekend, I'm out there with my camera, my field notebooks, and usually a thermos of cold coffee. And about 2 years ago, I remember this very clearly because it was the week after Thanksgiving. I drove to the Cascades with my friend Danny, a nurse who has tolerated my mushroom obsession with varying degrees of patience. I was crouched next to a pine tree when I found something that cut me off mid-sentence before I could even finish my thought. It looked like a small white pincushion, soft and pale and almost innocent-looking, except from the top it was weeping deep red droplets like it had been stabbed. Danny said, and I quote, "I need you to know that I am not touching that." And honestly, reasonable reaction. But I couldn't stop thinking about it.
That, right there, is Hydnellum peckii, the bleeding tooth fungus, also called the devil's tooth, also called the strawberries in cream mushroom, which feels optimistic to me. Some people also call it the red juice tooth, and it is one of the strangest, most visually striking fungi on the planet. But here's where it gets interesting. Researchers studying this mushroom's chemistry found inside it a compound called Atromentin. And Atromentin has shown anticoagulant properties. It can prevent blood from clotting. But more than that, and this is the part that made my jaw drop when I first read the research, it has shown neuroprotective effects in early-stage laboratory studies, meaning it may help protect nerve cells, the kinds of nerve cells that Alzheimer's destroys. Now, before we go further, I want to be straight with you. We are talking about early-stage research, not a cure. But the science is real, it's published, and it's being taken seriously. And the story of how this compound was found and what it could mean is worth your full attention. So, let's go deep on this. Let's talk about what the bleeding tooth actually is, where it lives, what's inside it, and what scientists are genuinely hoping it might do for the human brain. Grab your coffee, leave a comment, like, and subscribe. This is going to take a while. Let's start with the basics, because I think a lot of people hear bleeding tooth fungus and assume it must be some rare, exotic, tropical species, like something out of the Amazon. Nope. Hydnellum peckii grows in conifer forests, especially pine forests across North America and Europe. If you're in the Pacific Northwest like me, it's actually not that hard to find. It prefers well-drained sandy soils near spruce, fir, and pine in Oregon, Washington, up into British Columbia. It's also been found in the UK, Germany, Sweden, Korea, and parts of Iran. The name Hydnellum comes from the Greek hydnon, meaning truffle or earth mushroom, and peckii is named after Charles Horton Peck, a 19th century American mycologist from New York who spent decades cataloging fungi and seems like someone I would have loved to grab coffee with. But here's the thing about Hydnellum peckii that distinguishes it visually from almost anything else you'll find on a forest walk. Young specimens ooze a deep red or pinkish liquid from their pores, not from a wound, just naturally, through a process called guttation. Let me explain that, because it's actually wild. You know how sometimes you wake up in the morning and there are water droplets on grass, even when it hasn't rained? A lot of people assume that's dew. Sometimes it is, but sometimes those droplets are being pushed out from inside the plant. The plant's root pressure forces water up through the tissue, and it just seeps out through tiny pores called hydnothodes. That's guttation. Fungi can do a version of this too, and Hydnellum peckii produces a pigmented liquid through its pores that contains, among other things, that Atromentin compound I mentioned. So that red bleeding you see, that's not damage, that's the mushroom doing exactly what it's supposed to do. Think about that for a second. The thing that makes this mushroom look like a crime scene is actually a feature, not a bug. The first time I properly identified one, not just the time I found it with Danny, but later when I sat down and really studied it, I was up near the Diamond Peak Wilderness, about an hour southeast of Eugene, late October, maybe 3 years ago, overcast sky, that kind of flat gray light that makes colors pop in the forest. I was actually looking for chanterelles. I had my basket, I was feeling optimistic, and I found the chanterelles, a beautiful flush of them, under a big Douglas fir, but about 20 feet further on, I nearly stepped on what looked like a melting snowball with red paint splattered on it. I crouched down, pulled out my hand lens, and the more I looked at the underside of this thing, the more fascinated I got. Because here's another thing about Hydnellum peckii that distinguishes it from a lot of mushrooms. It doesn't have gills. It doesn't have pores like a bolete. Instead, the underside is covered in tiny, densely packed spines, little downward-pointing teeth, almost like a hairbrush. This is why it belongs to the group called tooth fungi, or hydnoid fungi.
That spiny underside is the spore-bearing surface, and functionally, it works. Spores fall freely from the tips of those teeth and get caught in air currents. It's elegant, actually. Weird-looking, but elegant. Now, here's something that surprised me when I first learned it. Hydnellum peckii is not edible, but it tastes absolutely terrible. Intensely bitter, almost medicinal. No one eats it. Traditional foragers across its range basically ignored it, which is actually kind of ironic, given what we now know might be inside it. And it's also not rare in the sense of being endangered, though populations are declining in parts of Europe due to nitrogen pollution, which affects the fungi's relationship with tree roots. But we'll get to that in a minute. The mature specimens look completely different from the young ones, by the way. That's something that trips people up. Young Hydnellum peckii, those are the ones that bleed. They're white or cream-colored with those vivid red droplets. As the mushroom ages, it darkens dramatically. Old specimens are brownish-gray, almost black, and they don't bleed anymore. They look like a completely different species. I've had people argue with me about this in the field. This one guy at a local foray I helped organize, he must have been in his 60s, very confident, was absolutely certain that an old dark specimen was a different mushroom entirely. Not Hydnellum peckii, he said, because that one bleeds. I had to pull up 3 different sources on my phone to convince him. He eventually came around, I think. He left without saying much. Forays are interesting social environments. Okay, so we've established what this mushroom is, where it lives, what it looks like, Now let's get into the chemistry, because this is where things get genuinely exciting. Hydnellum peckii produces a suite of chemical compounds, and they were largely unknown to Western science until the late 20th century. The key ones are Atromentin. This is the big one, a quinone pigment, and if that word means nothing to you, don't worry. It just means a specific type of organic molecule with a particular ring structure. Atromentin is actually what gives the bleeding liquid its red color. It's been found in several fungi, but Hydnellum peckii produces it in notable quantities.
Thelaphoric acid, also a pigment, related to atromentin, deep red to purple-colored. These are the ones that have attracted particularly significant attention for their potential biomedical applications. Now, here's the thing about atromentin that blew my mind when I first read the research paper. I was sitting at my kitchen table at like 11 at night, drinking the tail end of a pot of coffee, and I had to reread the passage 3 times. Atromentin's molecular structure bears a striking resemblance to heparin. And heparin, if you don't know, is one of the most widely used anticoagulants in medicine. It's been used since the 1930s to prevent blood clots in surgery patients, in people with certain heart conditions, in dialysis. And here is a fungus producing something structurally similar, naturally and in a forest. I remember sending that paper to my friend who's doing a PhD in biochemistry at University of Oregon. His response was a voice memo that was mostly him saying, "Wait, hold on, wait," repeatedly for about 30 seconds before he actually formed a sentence. That's the reaction of someone who understands the implications. Whenever he does the "wait, hold on" thing, I know I've found something worth paying attention to. But let me be careful here and give you the full picture, because this is science, and science is complicated, and I've watched enough bad health content on YouTube to know exactly how easy it is to go from promising preliminary research to "this mushroom cures everything." Atromentin shows anticoagulant activity in vitro, meaning in lab conditions, in cell cultures and test tubes. The jump from that to human clinical application is enormous. There are questions of bioavailability. Can the human body actually absorb and use this compound the way we'd want it to? There are questions of dosage, of toxicity at higher concentrations, of delivery mechanism. These are not small questions. And yet the research continues. And the reason it continues is that the initial findings are genuinely interesting enough to pursue. So how does any of this connect to Alzheimer's disease? Let me give you a quick, and I mean quick, I'm not a neurologist, overview of what Alzheimer's actually is, because I think most people have a kind of fuzzy understanding of it, and the specifics matter here.
Alzheimer's disease is the most common cause of dementia. It accounts for somewhere between 60 and 80% of dementia worldwide. Right now, more than 55 million people are living with dementia globally. In the United States alone, the Alzheimer's Association estimates that more than 6.9 million Americans aged 65 and older are living with Alzheimer's disease. That number is expected to nearly double by 2060. And we still don't fully understand what causes it. Let that sink in. This is a disease that affects tens of millions of people, that has been studied for a century. It was first described by Alois Alzheimer in 1906, and we are still arguing about the mechanisms. What we do know is that the brains of Alzheimer's patients show characteristic changes. There are abnormal protein deposits, amyloid plaques between neurons, and tau tangles inside neurons. There's a progressive loss of synaptic connections. The brain literally shrinks. The dominant hypothesis for a long time was the amyloid hypothesis.
the idea that those amyloid plaques are the primary driver of the disease and that clearing them should be the goal. Pharmaceutical companies have spent billions of dollars chasing this. There's genuine, cautious optimism there. But, and this is a big but, clearing amyloid hasn't always translated into meaningful clinical improvement, which suggests the picture is more complicated. Maybe amyloid is a symptom as much as a cause. Maybe inflammation is doing more damage than we thought. Maybe there are multiple disease processes happening simultaneously. And that's where things like Atromentin start to become interesting. I want to take a second and get personal here, because I think this matters. My grandmother, my dad's mum, Emma, was diagnosed with Alzheimer's when I was 19. I watched what happened over the next 7 years, and I can tell you that there is no way to fully prepare yourself for watching someone you love lose themselves, piece by piece. She forgot who I was when I was 23. She forgot who my dad was a year later. She passed when I was 26. I'm not telling you that to make you feel sad, well, maybe a little, but to tell you why this topic isn't just an intellectual exercise for me. When I read about researchers finding neuroprotective compounds in a mushroom I found in an Oregon forest, I feel that in my chest in a way that's hard to articulate. It matters. This work matters. So, neuroinflammation.
Recent research has increasingly pointed to chronic inflammation in the brain as a major player in Alzheimer's progression. And this is where Atromentin has attracted interest. In laboratory studies again, in vitro, we're not claiming anything beyond that, Atromentin has shown effects that suggest it might help inhibit certain inflammatory pathways in neural tissue. But separately, its antioxidant and anti-inflammatory properties are what the neurological researchers are paying attention to. A 2021 paper from a team in Europe looked at the neuroprotective potential of several bioactive compounds from tooth fungi, including from the Hydnellum genus. Their conclusion was careful, as good science should be. The results were promising and warranted further investigation. We're not saying we've found the cure, but at least it's promising and worthy of follow-up. That's how science actually works. It's not a series of dramatic revelations. It's a slow accumulation of evidence, most of which reads like, "We found something interesting and need to look at it more carefully." I'll be honest, sometimes I find this incredibly frustrating. There have been like 4 different studies in the past decade showing fascinating potential in various fungal compounds for neurological conditions, and they all end with further research is needed, and then you don't hear anything for 3 years. My friend Danny, the nurse I mentioned earlier, has a saying about this. She says medical research moves at the speed of making sure we don't accidentally kill anyone, which is like a good principle, but it's hard when you're waiting. Okay, let's zoom out a little bit, because to fully appreciate Hydnellum peckii, you need to understand something about its relationship with trees, and this is actually one of my favorite things in all of mycology.
Hydnellum peckii is mycorrhizal. That word mycorrhizal comes from the Greek for fungus, mykos, and root, rhiza. A mycorrhizal fungus forms an intimate association with the roots of trees and other plants. The fungus wraps around or even penetrates the root cells, and the 2 organisms exchange nutrients. The tree photosynthesizes and produces sugars. It shares those sugars with the fungus. The fungus, in return, dramatically extends the tree's ability to absorb water and mineral nutrients, especially phosphorus and nitrogen from the soil. The fungal mycelium can extend meters from the tree roots, effectively acting as a vastly extended root system. This is why Hydnellum peckii populations are declining in parts of Europe. Nitrogen deposition from air pollution, primarily from agriculture and vehicle emissions, changes the chemical balance of the soil.
The tree has less need for the fungus's help with nitrogen acquisition, so the relationship weakens. The fungus suffers, which is actually a pretty bleak metaphor for what happens when you make something less necessary.
I have this very clear memory from a few years ago. I was hiking with my dad in the Cieslaw National Forest. He's not a mushroom person at all, he's more of a bird watcher. But I was trying to explain mycorrhizal networks to him, and I could see his eyes starting to glaze over. So I stopped mid-trail and I said, "Dad, you see that big Douglas fir?" He said, "Yes." I said, "Right now, underneath your feet, that tree is trading food with that mushroom over there. And in exchange, the mushroom is basically acting as an extra set of roots." And he looked down at the ground, and then he looked up at me, and he said, "So the whole forest is just connected?" And I said, "Yeah, kind of." And he was quiet for a minute. And then he said, "That's the coolest thing you've ever told me," which felt like a win. Carbon, nutrients, and even warning signals can pass through fungal networks between individual trees. If an insect attacks one tree, the chemical distress signal can travel through the fungal web, and neighboring trees can begin mounting chemical defenses before the insects even arrive. This is the fungi doing something that blurs the line between competitor and collaborator, between organism and ecosystem. And Hydnellum peckii, by being mycorrhizal, is woven into this network. When you find it in a forest, you're not just looking at a mushroom, you're looking at the visible fruiting body of an organism whose mycelial threads extend through the soil in all directions, touching, connecting, trading with the trees that surround it. That red bleeding surface you see, that's maybe 5% of the actual organism. The rest is invisible, underground, doing its work. I think this is part of why fungi, and this mushroom in particular, deserve so much more attention than they get. We have this instinct to look for the spectacular and obvious, the big whale, the bright bird, the striking flower, and we completely miss the vast, ancient, complex network of life happening right under our feet. My neighbor's daughter, she's 9, her name is Rosie, and she sometimes comes with me on short walks when her mum is okay with it. Last fall, I showed her a section of old mycelium, just that white cottony network in a decomposing log, and explained that it was alive and that it was eating the wood. She looked at it for a really long time. Then she very seriously said, "Is it eating everything?" And I said, "Not everything, but it's trying." She backed away from the log a little bit. I probably could have phrased that better. But she's been interested in fungi ever since. Here's something I want to put on the table because I think it's important context for the bleeding tooth story.
The history of medicine and fungi is long, and we have not been paying enough attention to it. Penicillin, the antibiotic that has saved more lives than almost any other medical discovery in human history, comes from a mold, Penicillium notatum. Alexander Fleming didn't discover it in 1928, so much as notice it had been there all along, growing on a contaminated petri dish, killing bacteria. Some historians estimate penicillin has saved over 200 million lives since it was isolated into a usable medicine in the 1940s. 200 million lives from a mold that nobody was paying attention to. I think about Fleming a lot, actually. Like the version of events where he just throws away the contaminated petri dish, which is what most scientists would have done, because a contaminated experiment is a failed experiment. But he noticed something unusual, and he didn't dismiss it. He sat with the weirdness. I try to remember that when I'm in the field, and something catches my eye in a way I can't immediately explain. Cyclosporin, the immunosuppressant drug that made organ transplantation possible, that's allowed millions of people to receive kidney, heart, and liver transplants comes from a soil fungus called Taliaplicadium inflatum. It was discovered in the 1970s from a soil sample collected in Norway. Lovastatin, one of the first statin drugs used to lower cholesterol and prevent cardiovascular disease, was originally derived from Aspergillus terreus, a fungus. Ergotamine, used to treat migraines, comes from the ergot fungus. The list goes on, and it keeps getting longer as researchers look more carefully. Now, lion's mane, Hericium arenaceus. I have to mention this because it's the most discussed fungus in the neurological research space right now, and comparing it to the bleeding tooth is instructive. Lion's mane is another tooth fungus, interestingly, another hydnoid species, and it's been the subject of considerable research for its production of compounds called hericinones and arenasins, which have been shown to stimulate the synthesis of nerve growth factor, NGF, a protein that promotes the survival and growth of neurons. There have been small human trials, genuinely small, but actual human trials, showing some cognitive benefits in older adults with mild cognitive impairment.
I take a lion's mane supplement most mornings. I want to be transparent about that. Do I know with certainty it's doing anything? No. The evidence for human cognitive benefits is still preliminary, but it's not harmful. The biological mechanism is plausible, and honestly, it tastes good in coffee. My girlfriend thinks I'm a fungus evangelist. She's not entirely wrong. The reason I bring up lion's mane in the context of the bleeding tooth is that it shows a proof of concept. Tooth fungi, the hydnoid group, seem to be a particularly rich source of bioactive neurological compounds. That's not coincidence. These organisms have been engaged in chemical warfare and chemical cooperation with their environments for hundreds of millions of years. They produce compounds with extraordinary precision and specificity. We are just beginning to understand what those compounds can do. I have this analogy I use when explaining this to people. Imagine the entire fungal kingdom is a library, and each species is a book. For most of human history, we've only read maybe a few hundred books out of a library that contains somewhere between 2 and 5 million volumes. The estimates of total fungal species on Earth range from 2.2 million to 3.8 million, and only about 150,000 have been formally described. We are profoundly, almost embarrassingly ignorant of what's in that library. And every time we actually open a new book, like the bleeding tooth, like lion's mane, like the fungi they found growing toward the radioactive waste at Chernobyl, actually feeding on nuclear radiation, it's like discovering the library has entire wings we didn't know existed. So, where does the bleeding tooth mushroom stand in the research landscape right now? Honestly, it's still early days. The most active research into Hydnellum peckii compounds has been coming out of European institutions, particularly in Scandinavia, Germany, and the Czech Republic, where the mushroom is relatively well studied. There are researchers in South Korea who have also published on it. In the US, it tends to show up more as part of broader surveys of hydnoid fungi bioactivity. The Atromentin and scabrosin research is the most promising thread. Scabrosin esters, in particular, have shown cytotoxic effects against certain cancer lines, not just the neurological angle. This is a compound that is doing multiple interesting things simultaneously. Here's something I've come to understand through years of following this kind of research. The problem is usually a lack of interesting compounds. The problem is funding. Medical development is extraordinarily expensive. Running clinical trials costs hundreds of millions of dollars. A compound derived from a wild mushroom that nobody can patent in its natural form doesn't have the same commercial incentive driving investment that a synthesizable novel molecule might. This is a genuine tension in medical research that I don't have a good solution to.
It's why organizations like the American Mycological Society or academic researchers applying for NIH grants are important. They're doing work that market forces alone wouldn't prioritize. There is also genuinely promising work happening in mycoremediation and material science with these fungi, but that's almost a separate documentary. The short version is that some of the same organisms with interesting biomedical chemistry are also showing potential for breaking down pollutants and even plastic waste. I keep coming back to the Chernobyl story because it just won't leave my brain. In the years after the disaster, researchers found that not only had certain fungi survived the intense radiation, they were growing toward the radioactive particles, as if feeding on the radiation itself, using it the way plants use sunlight. It's called radiotrophic fungi, and it is one of the most extraordinary things I have ever learned. It suggests that fungi have metabolic capabilities that we haven't even begun to catalogue properly. And if a fungus can adapt to literally eat nuclear radiation as an energy source, what else might these organisms be capable of that we haven't found yet? I'll get into these in later videos. Let me bring this back to the core question. Can the bleeding tooth mushroom actually help fight Alzheimer's disease? The honest answer right now is we don't know. What we do know is 1. It produces compounds that have demonstrably interesting biological activity. 2. Some of those compounds have shown neuroprotective effects in laboratory conditions. 3. The mechanisms by which they operate, primarily anti-inflammatory and antioxidant pathways, are genuinely relevant to Alzheimer's disease biology. 4. Researchers continue to study it, which means they find the data compelling enough to keep pursuing. That's not a cure, but in the language of scientific research, that's actually meaningful progress. The history of medicine is full of discoveries that started with someone saying, "This is interesting, let's look closer." Penicillin started with a contaminated petri dish. Statins started with a soil sample from Norway. The bleeding tooth mushroom may start with a hiker stumbling across something that looks like it escaped from a horror movie in an Oregon forest.
I want to circle back to where we started. Alzheimer's disease, the gradual disappearance of a person, the slow erasure of every memory, every face, every thread of connection to the life they built. I think about my grandmother Emma sometimes when I'm in the forest, about what she would have made of all this, of mycology, of these strange organisms that live in the dark and do things we're only beginning to understand. She was a practical woman, a gardener. She would have wanted to know if it actually worked. The last real conversation I had with her, a conversation where she knew who I was and where we were, she told me she was afraid. She said she could feel herself getting confused more easily. She knew what was coming. She'd watched her own mother go through it. She held my hand, and she said, "I hope they figure it out before it gets to you." She was thinking about me even then. That's the kind of person she was. I don't know if the bleeding tooth mushroom is going to be part of figuring it out. Maybe Atromentin will turn out to be a dead end. Maybe it'll lead to something. Maybe there are 50 more compounds in that red ooze that nobody's identified yet. Maybe the answer isn't in the compounds at all, but in understanding the mechanism, the pathway that Atromentin interacts with, and that leads to something synthetic that works better. Science doesn't move in straight lines. It moves in spirals. It comes back around to things it dismissed, finds new angles, surprises itself. I was out last autumn, late November, and I sat down on a log, a big decomposing cedar, and just stopped, which I don't do enough. Usually I'm moving, looking, cataloguing, but I just sat and looked at the forest floor,
and thought about everything that was happening in the inches below me, the mycelial networks, the chemical signals, the hundreds of millions of years of evolution that produced all of this, all at once, all silently. And I felt genuinely something like reverence. Not religious, not mystical, just a deep, overwhelming sense that we are surrounded by complexity we cannot fully see, and that occasionally we get to lift the corner of it and catch a glimpse of something extraordinary. The bleeding tooth mushroom is one of those glimpses. It's strange and a little bit ugly, and it looks like something out of a nightmare. It tastes terrible and you can't eat it, and it grows in the dirt beneath pine trees where most people would never think to look. And inside it are compounds that might one day contribute to protecting the human brain.
I love that. I love that the universe hides things like that in places like that. I love that the answer to one of medicine's hardest problems might be sitting in a forest somewhere in Oregon or Sweden or South Korea, waiting for someone to crouch down and look at it properly. So the next time you're out hiking, and I know not all of you hike, but maybe this will make you want to look down once in a while. Pay attention to the forest floor. The giant whale gets the documentaries, the lions get the documentaries, but underneath your feet there is an entire kingdom that has been alive and thriving and doing extraordinary things for 1 billion years longer than any animal that has ever existed. And some of it might just help save your brain. Thank you so much for watching this far. Seriously, a 40-minute video is a commitment, and I don't take that lightly. If this sparked something in you, curiosity, questions, disagreement, I want to hear it. Drop it in the comments. I read everything. A few things before you go. If you want to go deeper on the research, the Atromentin studies, the lion's mane trials, the mycorrhizal network literature, I've put together a reading list in the description. Everything I referenced in this video is linked there. Primary sources, not just pop science summaries. If you're new here, this channel is about fungi, forests, and paying attention. I go out into the woods, I film what I find, I explain the science as honestly as I can without pretending I know things I don't. Sometimes, when I can't find the right mushrooms, I use AI to create my dream shots with a real reference image. People are still a bit strict about this, but unfortunately I don't yet have the budget for high-end productions. Subscribe if that sounds like your kind of thing. I haven't been able to upload videos regularly lately, but new videos most Saturdays. And if you know someone who's been affected by Alzheimer's, who's watching their parent or partner or grandparent disappear, share this with them. Not because I want to give false hope, but because I think there's something genuinely meaningful in knowing that people are out there in forests and labs looking at weird bleeding mushrooms and asking what they might offer us. That work is happening. People are paying attention. Science is slow, but it is moving. And sometimes, the most extraordinary things come from the most unexpected places. I'll see you next week, and in the meantime, go touch some moss.